Hello everyone, welcome back to another video. My name is Saurabh Pawar. Today video topic is DNA replication. DNA replication is the process of DNA makes multiple copies of itself. It is biological polymerization which proceeds in the sequence of intention elongation, termination. It is enzyme catalyzed reaction. DNA polymerase is the main enzyme in the DNA replication process. Following are the some important steps involved in the DNA replication. First is initiation. DNA replication demands a higher degree of accuracy because even a minute mistake would result in mutations. Thus, replications cannot initiate randomly at any point in DNA. For replication to begin, there is a particular region called the origin of replication. This is the point where the replications originates. Replications begin with the spotting of the origin followed by the unwinding of the DNA strands. Unzipping of DNA strands in their entire length is not feasible due to high energy input. Hence, the first replication fork is created catalyzed by the helicase enzyme which unzips the DNA strand. Second step is the elongation. As the strands are separated, the polymerase enzyme start synthesizing the complementary sequence in each of the strands. The parental strands will act as a template for newly synthesizing daughter strands. It is to be noted that elongation is unidirectional, that is, DNA is always polymerized only in 5 to 3 directions. Therefore, in one strand, on other strand, it is discontinuous replication. They occur as fragment called Okazaki fragment. The enzyme called DNA lignase joins them later. Termination. Termination of replication occurs in different ways in different organisms. In E. coli like organisms, chromosomes are circular and this happen when the two replication forks between two terminals meet each other. Next one is role of enzymes in DNA replications. DNA replication is highly enzyme dependent process. There are many involved many enzymes involved in DNA replication, which includes the enzymes, the DNA dependent DNA polymerase, helicase, lignase, etc. Among them, DNA dependent DNA polymerase is the main enzyme. DNA replication process in prokaryotes. The DNA replication in prokaryotes take place in the following place. The two strands of DNA unwind it at the origin of replication. Helicase opens the DNA and the replication forks are formed. The DNA is coated by the single strand binding proteins around the replication fork to prevent rewinding of DNA. Topoisomers prevent the supercooling of DNA. RNA primers are synthesized by primase. These primers are complementary to the DNA strand. DNA polymerase 3 starts adding nucleotides at the end of the primers. The landing and lagging strands continue to elongate. The primers are removed and the gaps are filled with DNA polymerase first and sealed by lignase. DNA replication in eukaryotes. DNA replication in eukaryotes is similar to the DNA replication in prokaryotes. However, the initiation process is more complex in eukaryotes than prokaryotes. In eukaryotes, there are multiple origins of replication present. A pre-replication complex is made with other initiator protein. The process is entirely the same, but the enzymes used are different. Example eukaryotes, the polymerization process is carried out by the enzyme Pol6, where in prokaryotes it is done by DNA Pol3. Thank you.